serial dilution for hemagglutination. A simple dilution is one in which a unit volume of a liquid material of interest is combined with an appropriate volume of a solvent liquid to achieve the desired concentration. The dilution factor is the total number of unit volumes in which your material will be diluted. The dilution material must then be thoroughly mixed to achieve the true dilution. For example, a 1:5 dilution entails combining a 1 unit volume of solute, the matter to be diluted, plus 4 unit volumes of the solvent medium. Therefore, 1 plus 4 equals 5, which is then the dilution factor. For example, frozen apple juice concentrate is usually diluted with 4 cans of water, which is the dilution solvent, producing a dilution factor of 5. Therefore, the apple juice concentrate is now distributed through 5 unit volumes. This is a 1:5 dilution since the apple juice is now 1/5 as concentrated as it was originally. Serial dilution. Serial dilution is the stepwise dilution of a substance in solution. Usually the dilution factor at each step is constant. Serial dilution results in a geometric decrease of the concentration in a logarithmic fashion. The source of dilution material for each step comes from the diluted material of the previous. Twofold dilutions. A standard dilution technique used in the serology laboratory is the twofold dilution. Each tube will contain half serum and half diluent, which is usually saline. The first tube of the twofold dilution series contains the undiluted patient's serum. The second tube contains half the amount of serum and therefore half the amount of antibody. The third tube contains a quarter of the amount of antibody. The fourth tube contains an eighth. The fifth contains a 16th. The sixth a 32nd. The seventh tube a 64th, etc. A substitution of methylene blue for the patient's serum is made for this demonstration. Place 10 clean test tubes in a test tube rack. Add 1 ml of 0.9% saline to tubes 2 through 10 using a 5 ml pipette. Add 1 ml of methylene blue to tubes 1 and 2. With the same pipette, mix the contents of tube 2 by drawing the contents up into the pipette. The process of drawing up and dispensing out is considered to be mixing. To mix thoroughly, this should be repeated a few times. Note that tube 1 contains undiluted methylene blue, whereas tube 2 has half the amount of methylene blue, since tube 2 also contains 1 ml of saline. There is 1 part of undiluted methylene blue and 1 part of saline, which equals 2 parts or 2 volumes. Using the same pipette used to mix the contents of tube 2, transfer 1 ml from tube 2 to tube 3, which also contains 1 ml of saline. Mix contents of tube 3 well, as previously noted, by drawing up and dispensing out the contents of the tube. The half quantity of tube 2 has been diluted to 1/4 in tube 3, which is a 1:4 dilution. With the same pipette, transfer 1 ml of the well-mixed contents from tube 3 to tube 4, which then represents a 1:8 dilution. Continue to transfer 1 ml from tube 4 to tube 5, from tube 5 to 6, etc., to the last tube 10. Again, mix the contents of tube 10 well and discard 1 ml from that tube. Each tube contains 1 ml total volume. In looking at your serial dilution, you are able to see a decrease in the intensity of the blue color from tube 1 to tube 10. Tube 1 is undiluted and tube 2 is the first in the series of dilutions, which is a 1 to 2, followed by 1 to 4, tube 3, 1 to 8, tube 4, 1 to 16, tube 5, 1 to 32, tube 6, 1 to 64, tube 7, 1 to 128, tube 8, 1 to 256, tube 9, and 1 to 512, tube 10. Setting up a serial dilution is a much easier method to evaluate a variety of concentrations than calculating and preparing each dilution individually. The serial dilution of antibody is used to determine the amount of antibody present, which may be referred to as the semi-quantitation of antibody. This will be illustrated in the laboratory by an agglutination reaction, where there is a serial dilution of the antibody interacting with a constant concentration of the antigen. The following materials will be used: a 5% suspension of sheep erythrocytes, which is the antigen, undiluted rapid anti-sheep erythrocyte serum, which is the antibody, 0.9% saline solution, which is the diluent, and 3 1 ml serologic pipettes. Label the tubes from 1 through 5 and place in the rack provided. Add 0.4 ml of 0.9% saline solution to tubes number 2, 3, 4, and 5. The 1 ml pipette is divided into tenths and is referred to as a 2-deliver pipette. To dispense 0.4 ml of saline into the tube, you will draw the suspension up to the 0.6 ml mark on the pipette. Since at 0.9 ml, a tenth of a ml is dispensed. 0.8 ml, 2/10 of a ml is dispensed. 0.7 ml, 3/10 of a ml is dispensed. And at 0.6 ml, 4/10 of a ml will be dispensed. To draw up and dispel the saline, you will be using a pipette aid. Remove the plastic from the pipette, which assures the sterility of the pipette, and place the pipette aid securely on the pipette by rotating it a few times onto the top of the pipette to assure a tight fit. With a separate 1 ml pipette, remove 0.5 ml of anti-sheep serum and add 0.4 ml to tube number 1 and 0.1 ml of serum to tube number 2. Mix the contents of tube number 2 thoroughly by drawing up and dispensing out the contents of tube number 2 a few times. You want to make sure you mix well the contents of the tube, which contains 0.1 ml of anti-serum and 0.4 ml of saline. Tube number 2 is the first tube of the serial dilution and represents a final dilution of 1 to 5, where 1 represents the amount of antibody and 5 the total volume of the tube. Remove 0.1 ml from tube number 2 and place it in tube number 3. Tube number 3 therefore has a final dilution of 1 to 25, since 0.1 of a 1 to 5 diluted anti-serum was added to 0.4 ml of saline. Again, mix contents of tube number 3 thoroughly and transfer 0.1 ml of the suspension to tube number 4. Mix the contents of tube number 4 thoroughly and remove 0.1 ml from tube number 4 and discard the contents with the pipette in the biohazard bag. In a serial dilution, all the tubes should have the same volume. Tube number 4 has a final dilution of 1 to 125, since 0.1 ml of a 1 to 25 diluted antiserum was added to 0.4 ml saline. In this dilution, 1 to 5, 1 to 25 is called a 5-fold dilution. 5 times 5 is 25. 5 times 25 equals 125. Tube number 4 is the last tube of the serial dilution. Next, 0.1 ml of the 5% suspension of the sheep erythrocytes will be added to each of the 5 tubes. You need a homogeneous mix of the red blood cell suspension to be added to each tube. Prior to dispensing the sheep red blood cells, make sure you resuspend them using the 1 ml pipette. Draw up 1 ml of the 5% suspension and then dispense 0.1 ml into each of the 5 tubes. Discard the remaining 0.5 ml with a pipette in the biohazard bag. Shake the rack vigorously for 1 minute. Place your tubes into the tabletop centrifuge found on each lab bench. The centrifuge contains 4 carriers that each holds 5 tubes. Be sure to note that the arms of the carriers are numbered, so you will know which group of 5 tubes is yours after centrifugation. Safe centrifugation requires balanced loading of the tubes to be centrifuged. Balanced tubes are placed across from one another. Also, at the time of loading the centrifuge, make sure that no other tubes or materials are in the centrifuge, as they would be thrown around the chamber during centrifugation. Slide the lever on the centrifuge from the off to the on position. When the centrifuge reaches its maximum speed, wait about 30 seconds and then push the lever to the off position. When the carriers have stopped rotating, remove the tubes and place them in your test tube rack. You can see that after centrifugation, the cells are pelleted to the bottom of the tube. Tube number 5 is the negative control and will not have an agglutination reaction when resuspended. Tube number 1 will have the strongest agglutination reaction since you have undiluted antiserum. Gently shake the tube and look at the agglutinants, which cannot be resuspended to a homogeneous mix. Repeat this process for tube number 2 through tube number 4. You are looking for the last tube where a positive agglutination reaction is observed. In this serial dilution, it is seen as the lowest concentration of antibody, which is the highest dilution of the antibody, where agglutination is still visible. This will be recorded as the endpoint, which is the final dilution that was positive. The titer is the reciprocal of the endpoint. For this assay, the endpoint was 1 to 25, and the titer was 25.